To obtain organelle DNA, either isolate the organelle and then extract DNA, or obtain whole cell lysates, isolate DNA, and separate nuclear and organelle DNA. In isolating organelles to obtain their DNA, gently homogenize cells in an ice-cold isotonic solution to avoid breakage of the organelles by extreme shearing force, enzyme activity, or osmosis. Filtration then removes unbroken cells and cell debris. Next, centrifuge the filtrate at low speed to precipitate larger unwanted structures like the nucleus. Transfer the supernatant to a fresh tube and spin at higher speed, also optimizing duration to precipitate the organelle wanted. The separated organelle can then be lysed, the DNA extracted and purified. If organelle DNA is to be obtained from the cell lysate containing nuclear and organelle DNA, cesium chloride gradient centrifugation may be used to separate these DNAs. When solutions of cesium chloride are centrifuged at sufficient speed, cesium chloride forms a concentration gradient so that the solution at the bottom of the centrifuge tube is denser than at the top. The nuclear and organelle DNA present in the tube with cesium chloride will settle at different positions corresponding to their own density. Adding ethidium bromide enhances the density difference, separation, and visualization to aid in DNA collection. DNA is collected by inserting a needle at the appropriate location and removing the band which is most likely the organelle DNA wanted. The ethidium bromide and any cesium chloride present with the DNA needs to be removed, for instance by ion exchange chromatography or dissolving the ethidium bromide in N-butanol and removing the cesium chloride by dialysis. The DNA obtained can then be precipitated, washed, and resuspended in TE buffer.